All right, well, thanks, everyone. It's a pleasure to be able to host the panel on gene-modified therapies for the Alliance of Regenerative Medicine Investor Day. I'm Josh Wishamer. I'm one of the senior biotech analysts at Piper Jaffray. And gene therapy is a platform that we've become very interested in at Piper. The gene therapy field started over 2 decades ago. It did run into some early safety headwinds, but over time, these have been gradually addressed, and as the understanding of these technologies and the tools have advanced considerably. And the ability to use viruses or other gene delivery tools to change the genetic code of cells to replace lost function, to add new protein expression capabilities, has such broad applicability to countless diseases, including ones that have no current treatment options to date, as well as ones that do. And at Piper, we look at the enthusiasm and the interest that investors have expressed for the RNAi and the antisense companies based on those technologies' ability to knock down protein expression. And we consider the potential for gene therapy to be far greater. The fact is that the overwhelming majority of genetic disorders are loss-of-function mutations for which gene therapy is really a natural fit. As well, the early data that we've seen from a number of gene therapy programs, including the CAR T platforms, the chimeric antigen receptor T cells, and that's just one slice of the gene therapy pie, suggests that these therapies can not only reshape treatment of monogenic disorders, polygenic disorders, even liquid tumors, and potentially solid tumors. Now, it's hard to confine a discussion of gene therapy to just a 1-hour panel, but hopefully we can use this time to give a top-level overview of the field and its potential, then drill down into CAR T applications, which is an area of shared interest for our panel members. And with that, it's a good segue to introduce our panel members. To my immediate left, or from facing me, your right, we have Liz Wolfe from Sangamo Biosciences. Liz is the Senior Director of Corporate Communications. She actually joined Sangamo as a scientist and previously had worked at NIAID and the NIH. Beside Liz is Rob Ross from Bluebird Bio. Dr. Ross is VP of Clinical Development. He's also a medical oncologist, previously at the Dana-Farber Cancer Institute, where he still lectures, and he's spearheading much of the work on the CAR T program at Bluebird Bio. And then beside him, we have Dr. Bruce Levine from the University of Pennsylvania. Dr. Levine is the Barbara and Edward Neddard Associate Professor in Cancer Gene Therapy and Director of the Clinical Cell and Vaccine Production Facility at the Abramson Cancer Center and Perelman School of Medicine. Dr. Levine's gene therapy research interests include modification of T cells as part of CAR T programs, as well as for treatment of HIV. And University of Pennsylvania has really played a key role for many years now in advancing the field of gene therapy persistently and successfully. So first of all, welcome to all our panel members. Thank you for joining. And Bruce, why don't we start with you? Gene therapy as a platform's come a long way over the past couple of decades. UPenn has been right in the middle of the evolving field. If you could summarize the last 20 years of progress in about 3 minutes,
So
or less,
years and,
maybe
3
give us.
minutes, if I do my math, that's about 6 months per word. And if we look at the first use of genes in humans, it was in 1990 in 2 studies at NIH, the Rosenberg group, and also Mike Blaze and French Anderson and colleagues. And at the time, there was the use of gene therapy for a few general indications. One is gene replacement, and that was SCID-ADA, to provide a correct copy of the adenosine deaminase gene. And the other was to mark T cells, and there was thinking in the gene therapy arena primarily centered around, let's call it replacement or correction of a gene. Well, throughout the 90s, those studies continued, but at a very slow pace. And in the late 90s, we began our first work in gene therapy with what we believe was the first chimeric antigen receptor clinical trials with cell genesis, redirecting T cells to HIV-infected cells. Then we moved to another phase of gene therapy, and my colleagues can speak also to different uses of gene therapy. And I'm speaking primarily about gene-modified cells. And we think about providing a corrected gene. We think also now in the CAR-T platform about redirecting T-cell specificity to find cancer and kill cancer. When we think about that and we think about the history of the development of gene-modified cells, there has been a lot of hills and valleys. And I think in the earlier discussion, there was mention of gene therapy death that occurred 14 years ago. There was great success with correcting ADA, and there was great success in larger cohorts of patients in trials in England and in France in kids with severe combined immunodeficiency. It was later found out that by gene modification of stem cells, there were some serious adverse events. However, to frame that in context, kids with this severe immune deficiency disease face a bleak prognosis, especially if they don't have a stem cell donor. So in the end, somewhere around 12 or 14 kids were treated. 4 developed leukemia, 1 died from their leukemia, others were successfully treated. So that is a peak and a valley, if you will, and we're to the point now in the mid to later 2000s where we began working on the chimeric antigen receptor T-cell technology. And we published 3 patients in New England Journal of Medicine in August 2011. And it's difficult to convey the amount of attention that those 3 patients received. There were 800 media reports around the world. So why was that? We had 4 to almost 8 pounds of tumor disappear over a matter of weeks in these patients. They were extraordinarily well studied. We received attention from entities of all sizes to license that technology. And as you know, that was licensed by Novartis. And I think what that drove home is it was a seminal moment in the field of gene therapy. We no longer thought of it as a boutique therapy with the investment of large pharma. And we see that also validated with Celgene and Bluebird and Juno and Kite Pharma, and you can go on in the CAR T-cell field. And before I wind up and pass it to my colleagues, I'll also mention that we've been doing HIV gene therapy. I mentioned the CAR studies in HIV. We've worked with small biotechs, Virixis, and we've worked, full disclosure, with Sangamo for a decade now, and the results of that first clinical trial using zinc finger nuclease to modify the genome and engineer HIV resistance, that was published 2 weeks ago, also in New England Journal of Medicine, and also received very wide play. I want to make one other point, and that is the public perception. And I think in the early days of the field, there had been a fear of gene modification, and you see that in the Recombinant DNA Advisory Committee, and you see that in the fear in some quarters of genetically modified organisms. And we were, I think, thunderstruck by the amount of attention that our gene therapy trials, the publications and the press coverage. And what that tells you is that the public is amenable to gene therapy in a therapeutic application. There is no fear. We've had thousands and thousands of patients and families contact us and want to be on our clinical trials. So we are past the point of the roller coaster, I think, and we're on the upswing in the terms of application of these technologies.
That's a great overview of I guess the history of the efforts that culminated in that New England Journal publication that really, I think, catalyzed the attention and the subsequent explosion in the interest. Liz, maybe from your perspective at Sangamo, you can help us understand what's been going on over the last decade plus. Have you seen other kind of catalyzing events similar to that New England Journal publication that have broadened the interest in gene therapy, not just for CAR-T, but beyond?
Well, I think I agree with Bruce, actually, the introduction or the coming into the field of a number of different companies and the successes that we've seen in the trials like the SCID trial, although that was a sort of a lumpy progress, in actual fact, as you point out, that was a huge success for the vast majority of those patients. And so I think that we're just seeing a growing awareness and a growing understanding that these types of therapies are, a good opportunity for us to start to think about genetic cures for diseases in a way that we couldn't in the past. And so definitely it's helped to have more voices in the field, and I think it's definitely helped to have large pharma interest as well in this space.
What has been the gating step for the progress, given how many years have been invested? What were the learnings along the way that kind of went from early interest to where it is now? And it's taken 14 years, I guess, plus for gene therapies to now start to explode. What took so long?
Do you want to try that?
So I think there are a number of hurdles throughout the potential development, and one thing that we don't want to miss is gene therapy is a broad tent, right? And the hurdles within the different potential therapeutic applications may not be the same. So the hurdles just around safety, which has clearly been a hurdle, and Bruce described some of it very nicely. The safety of instilling an adenovirus or an AAV vector directly into someone's body is a different safety hurdle than modifying someone's hematopoietic stem cells, which is then a different safety hurdle than what we see in the CAR T world. So clearly safety in general has been a hurdle, but it's not a generalizable safety concern. Is it an autoimmune reaction to a virus? Is it off-tumor on-target toxicity? Is it a concern, as it was with the SCID trials, is it a concern around leukemiogenesis in the setting of insertional oncogenesis, right? So I come at it from a clinical perspective. I think there are clear manufacturing concerns as well, which are clearly being overcome. The safety concerns are about learning in the clinic and then making incremental steps forward and getting better and better at it over time.
And for any of the panelists, to what extent has funding been a gating step? To what extent has lack of enough biologic understanding of pathways to know which targets to pursue been a limiting extent? Or really has the gating factor really been about safety and making sure that we're going slowly and doing this all right?
So I can speak to the funding a little bit, and that was when we began our CAR T program. We applied for a grant from a foundation, the Alliance for Cancer Gene Therapy. And you could think of this as venture philanthropy. This was to get a field started that at NIH study sections, there was a lot of difficulty, people viewing this new technology, not believing that it could be feasible or broadly applicable. So this foundation, and I hold the chair now, but this foundation has funded many investigators across the country doing basic, applied, and clinical gene therapy research. They really got us started, and we had also applied to the NIH, but got denied 3 times through funding by NIH. And now, fortunately, we have the Novartis Alliance, and that's not the concern to apply for NIH grants. But also point out that things have come back full circle, because today the NIH director, Francis Collins, is testifying in front of the House Appropriations Committee, and as part of his testimony, he's going to mention our CAR T program in one of our patients, and he's at the University of Pennsylvania. And then he's also going to testify to the Senate and talk about one of the children. So and I think that now NIH, at least the director gets it, but those forces are beyond our control, and there's very large policy and taxpayer and budget issues in there. I think to advance this field, we cannot rely on government funding. It will come from the private investment community, it will come from venture philanthropy, and it will come from biotech and pharma alliances with academia.
Liz, as a former scientist at the NIH, how can you possibly explain why the NIH did not fund the therapy?
Well,
I'm actually just, it's not actually,
I'm not sure
that's
I'm gonna
not the
take
real
that
question. At Sangamo, before we really drill into CAR T as a specific application of gene therapy, maybe you can give us a snapshot of what you are doing at Sangamo, how you think about prioritizing different programs and opportunities, because there is so much going on at Sangamo and beyond for gene therapy more broadly.
Sure, and we are a definite sort of subset of gene therapy. What we're doing at Sangamo is developing a powerful platform called zinc finger DNA binding proteins. And these proteins allow us to do a couple of different things because they act at the DNA level. So what we can do is we can affect gene regulation, so we can either regulate genes up or down, and we can also modify the genome. So this gives us a way to, in a very targeted and specific way, to either add a gene into a specific site, and we're doing this with, in collaboration with Shire in our in vivo protein replacement platform, where we're adding in transgenes to basically cure diseases that are currently served by enzyme replacement therapy, things like hemophilia A and B and lysosomal storage disorders. What we're using there is we're using the body's liver as a mini protein production factory. We're using the albumin locus, which is a very powerfully produced gene. We're using that promoter to drive the production of a gene of interest, factor VIII, factor IX, or one of the lysosomal storage diseases. The other thing that we can do is to knock genes out, and as we were talking, as Bruce was mentioning, we've got an active program in HIV where we're knocking out the CCR5 receptor, and this is an essential co-receptor for HIV entry. So it's a bit like taking the doorknob off the door so that HIV can't get into the cell. And the idea there is, and this is a common theme throughout gene therapy, it's basically enabling the body to do what it needs to do. So not essentially working with what you've got to make it better. So what we're trying to do is to generate a protected compartment of the immune system that can't be infected by HIV, so that these immune cells can help to control the virus in the same way that you would control with a viral infection like a cold or chickenpox or something like that. And so that's the idea. We've seen encouraging results. The first publication from this, and in fact the first in-man genome editing study was published in New England Journal of Medicine at the beginning of the month. And we've seen very encouraging results in both viral control and in control of the reservoir, which is a part of the HIV infection that doesn't get addressed by antiretroviral therapies currently. So small molecule drugs are really not touching the reservoir, and this is going to be the big problem that we have to tackle in order to see a cure for this particular disease or functional control of the virus. So what we're trying to do is in a number of different diseases, and we're focused on monogenic diseases initially, because that is really the most obvious target to go after. We can actually change a gene, we can add a gene in very specifically, so it makes sense for us to go to look for genetic cures for diseases that are caused by a mistake in a single gene.
I think what I find so exciting is that that's just the tip of the iceberg. You show success, you replicate the success. You've hit the nail with your hammer, and then you start looking around for other nails, and there are plenty of low-hanging fruit nails to hit across rare orphan diseases, and then more broadly in platforms that are very modular. So the learning from doing it once really informs your subsequent successes. Maybe we can dive in a little bit deeper into the CAR T approach, and Rob, help us understand what CAR T is. What is the concept behind it that we're executing on here?
Right. So exactly, yeah. So fundamentally, within a CAR T program, you are taking someone's normal T cells, having that T cell express a receptor that is going to identify and then become active when it encounters your tumor cell. That T cell will then perform its typical cytotoxic role. It will try to kill the T cell, but it will also bring in other elements of the immune system to try to create this immune response. And the power there is the power of the immune system, and it can be extraordinary. And I think in the work that's been done at Penn and at Memorial and at the NCI, you can see that patients can have dramatic responses in a short time frame, but that dramatic response can also be accompanied with toxicity that's also associated with overwhelming immune system stimulation. So in a nutshell, you're using the body's own T cells and giving it the ability to recognize a tumor cell. So you can see within that context, you need to understand what that target is in the tumor cell that's going to allow the T cell to recognize it. But then also you need to understand where that target is also expressed, because if that target is also expressed on normal tissue, the T cell can't distinguish between the tumor cell and a normal cell, and you can have an overwhelming, fundamentally autoimmune response that can also be life-threatening.
Bruce, what have been the lessons for how to do it right? What are the variables that you control that determine or determine the difference between failure and success for CAR T?
Well, I wish we knew all of them. We know some of them, which is the method to expand and generate T cells, the method to deliver the gene, and the method for the chimeric antigen receptor design. But I want to pick up from Rob and speak to how this is a fundamentally different drug. And I think Rob was getting to it, and that is the T cells divide. So you can think of this as a therapeutic vaccine with memory. It's fundamentally different dosing a drug that is a dividing drug from a drug or an antibody or even a cell like a dendritic cell that does not divide. We infuse these cells to the body, and they're dividing 1,000-fold or more over a period of weeks. So every cell that is infused can generate daughter cells and granddaughter cells that can find tumor, kill tumor, and then can provide a sentinel function for months or years. And we've observed that these cells traffic, they engraft not only to the blood, but also to the bone marrow and to the cerebral spinal fluid, which is a significant source of disease metastasis. So they can provide not only the initial therapeutic effect, but ongoing sentinel ability. And as far as how long these cells can last, the first CAR trial that I told you about in HIV conducted in the 90s, we got samples from those patients because they come back every year as part of FDA mandated guidance. And we showed that we thought we would see a drop-off after some months or years. And we showed in a paper in Science Translational Medicine a few years ago that there is a relatively steady persistence of those cells in the setting of HIV infection, albeit these patients are on drugs. And when you calculate the half-life of the first 9 years, these T cells have a half-life of 14 years. So, and this is very important. With one or a couple infusions, you can have protection, in theory, not only for weeks or months, but for years. So that, when we think about autologous therapies, which this is very important distinction from other types of therapies, even other types of regenerative and immune therapies.
Yeah, Bruce, it's a critical point. I think the upside is the surveillance function, right? And that, I think, has resulted, what is most extraordinary to me as an oncologist from the work that's been generated from University of Pennsylvania and others, it's not just the complete response rate, it is the duration of those responses. So with chemotherapy, we can get complete responses, but they tend often not to be durable. So it's the sentinel function, which is extraordinarily exciting. The flip side, though, is that it's hard to turn off, right? So it's not, you can't just stop giving the patient the drug. Now, there are ways of managing that, but you have to be aware and have plans for if there is toxicity, how are we going to mitigate or moderate that toxicity? Because it's not a classical safety efficacy standard.
And what are the solutions? How do you do that?
I can give you a couple of solutions, and Rob and Liz can add others. One solution that the group at Baylor and others have worked on is a suicide gene or a suicide switch, where you would add a drug that would kill those gene-modified T cells. Another solution that we've been working on is not using a lentivirus to deliver the gene encoding the chimeric antigen receptor, but to use RNA. And RNA is temporary. So we electroporate the RNA into the T cells. The chimeric antigen receptor is expressed on the surface, but it's only expressed for several days. So in the case of a lentivirus, we give one infusion or a couple infusions up front, and the cells can last a long time. With RNA-modified chimeric antigen receptor T cells, we give multiple infusions. And then if, as Rob says, you do see toxicity, you would just discontinue the next infusion, because those cells, as they divide, they will have less of the receptor on their surface, and the RNA and the protein itself will degrade. So that is a built-in safety mechanism. There is the suicide gene switch approach as well.
And maybe Liz and Rob, some of that technology now moves maybe a bit beyond your core competency in the gene delivery now into maybe using RNA and T cells. Is your view that, as companies, you will pursue the science based on CAR T and its potential, even if it pushes you beyond the core of gene therapy, or do you kind of look at your approach and say, well, we've got our toolkit and we're gonna go forward with our toolkit and let others try other approaches?
So we don't develop actually CAR T products ourselves, but one of the ways that we think that we can add value is in maybe the next generation of these products, where we can modify the genomes of these cells to either make them safer or more efficient, or to actually make them such that you could perhaps have an off-the-shelf type of product. And so that's where we see the value in our technology in this space. And as Bruce alluded to, one of the things that we do actually with all of our ex vivo, or one of the things we're certainly starting to do with all of our ex vivo administration of zinc fingers, because you only need the zinc finger nuclease to be there temporarily, we can deliver this with RNA. So that's a product that we've developed, that's a process that we developed with our HIV stem cell program, which is we're going to be filing an IND for that sometime around mid-year this year. And we're now transferring that program over as well to, or that process over as well to our T-cell. So our T-cell manufacturing for the HIV program. So, I think you go where the science takes you in these things. I mean, certainly you don't want to be sort of switching horses. We've got a technology which is amenable to delivery with any kind of vector. So we've been using adenovirus initially in our T-cell programs, and we've used adeno-associated virus in our in vivo programs. But certainly this RNA delivery is proving to be useful. So.
Yeah, and I think for us, clearly if not the core competency at Bluebird has been the ability to make high titer, high quality, high transduction efficiency lentiviral vector, right? So that is a pillar of what we do. And clearly lentivirus has been shown by Bruce's group and others, lentivirus has a clear role to play in the world of CAR T. But as we see these therapies develop, what's important for us is we take our own internal expertise and then have the appropriate collaborations and partnerships to leverage what we do well and leverage what others do well such that we can bring the most potential benefit to patients. And I think that's where the collaboration with Celgene and Baylor College of Medicine came from, which was trying to leverage all of our expertise to really push this forward. So.
I guess, Liz, you had mentioned off the shelf as a goal. Until we get to that point, and maybe Bruce, you can address this, is CAR T ready for prime time? What are the logistic hurdles? What are the manufacturing hurdles that we need to overcome to make this commercially viable?
Okay, so I'll take that one first. I think we are at a point where a lot of the science about making this work is known, and we have demonstrated therapeutic effect. We've treated about 80 patients now with the CAR lenti alone, more with the RNA and more with other approaches. So we're at a point where we can get this to work in a clinic. And how do we deliver this to hundreds and thousands? And I think right now, imagine a 1915 automobile assembly line or a car assembly line, right?
Yeah,
American Interceptor car assembly line. And it's very manual. There are people surrounding the car and they're making them one by one. And now imagine a 1985 assembly line, and you see the line and you see robots from either end making the welds and inserting parts. And it took about 70 years to get to that point. And I'll ask you to remember, if you were around, the early days of bone marrow transplantation, now called stem cell transplantation, in the 1970s. And what people were saying was that it would only be done at the Hutch and at Hopkins. That was boutique therapy that would never be widely available. And last January 2013, the 1 millionth stem cell transplant took place. And that took about 30 years. And I think we can have that again in the development of these cell therapies, even autologous therapies. And admittedly, we're at the early stages of developing the engineering to make this widely available. And I think tool makers like GE, Fenwal, others who are in the room and others who are not here, those companies have a huge role to play in developing the equipment and the instrumentation and also software to drive that, to be able to automate first pieces of this process and then more of this process to make it readily available. And we can't say that just because it's difficult, it shouldn't be done. We have the demonstration of the therapeutic efficacy, and it's our duty now to try and develop that and make it widely available.
Rob, is that a significant capital investment to build those capabilities? And is that something that ultimately may limit the number of entrants into this field?
So I think certainly the capital investment limits at the beginning, right? But I go back to something that Bruce hit on, which is if the clinical results are good enough, right, you are going to drive that investment, right? We have shown that this can be done reproducibly at major academic centers that do this well. Okay, again, Penn, Baylor, Memorial, the NCI, Fred Hutch. What we need to demonstrate now is that it can be done safely and efficaciously at the thousands of other cancer centers across the United States. And that's just considering the United States, not including Europe. If we can show it's reproducible and it can be done safely with appropriate parameters, then the outcomes will drive the investment.
I know for some investors who are familiar with some of the margin considerations for Provenge from Dendreon and how that's been a bit of a gating step for that product. Are we looking at ultimately a similar labor-intensive approach, or what's the feasibility of not running into that type of situation?
So I'm So one of the things I do is I take care of prostate cancer patients, and so Provenge is a story that I followed for a while from the clinical side. I don't think the analogy is very strong simply because one of the fundamental problems with Provenge, having nothing to do with the development plan, but having everything to do with the way it works, is that it's very difficult to determine on an individual patient basis who's benefiting. You can determine that on a population basis, but it's very difficult on an individual patient basis. Here, it is extraordinarily obvious to point out which patients are benefiting, and that's actually a theme for gene therapy in general, that we're not talking about p-values, right, which is a nice place to be when you're doing clinical development. Instead, we're talking about very obvious differences in response. So I think the uptake will be fundamentally different, in part because of the difference in the therapy.
And where are we seeing the CAR T approach have its greatest impact and across the liquid tumors so far? And what are there limitations? You just find a new tumor, find a new target, and deploy? Is that?
Yeah, so I'll just say for everyone who's not familiar with the target is CD19. It's present on B cells and B cell progenitors. It's not present on the hematopoietic stem cell, and B cell aplasia is an accepted toxicity with the use of Rituxan or rituximab. So we have treated patients with chronic lymphocytic leukemia and with acute lymphocytic leukemia. The first 3 patients were chronic lymphocytic leukemia. Our overall response rate is about 50%, a little bit under currently with the patients we treated. With acute lymphocytic leukemia, and both of these populations are relapsed or refractory, but acute lymphocytic leukemia, much more aggressive disease. 2/3 of the patients we have treated have failed an allogeneic stem cell transplant, some of them twice, and there's no option for them. I just saw update, and we're almost at 90% complete response rate. And if you'd asked me 5 years ago what would be our response rate, anyone would have been laughed out of the room if they said that. But we have the patients, they are responding. We have durable responders. The first 2 out of the 3 adult CLL patients we treated this summer will be 4 years out. The first child in 2 weeks will be 2 years out. So these are durable responses. As far as other hematologic malignancies, we're now treating patients with lymphoma. We've treated a patient, and we will have a trial opening in myeloma. And as I mentioned earlier, we're moving into solid cancers with the RNA approach and some in the lenti approach with pancreatic cancer, mesothelioma, breast cancer, and glioblastoma. So the targets are different in the solid tumors, and then there are considerations, as Rob said, with the specificity and whether we use a lentivirus or an RNA.
I mean, the hurdle, the data in B-cell malignancies is really extraordinary. Solid tumors represent a hurdle not just from a target selection standpoint, which is critical, but also from a tumor microenvironment standpoint. So as is highlighted by the successes of the anti-CTLA-4 and anti-PD-1 antibodies, but the tumor develops pretty extraordinary immune suppressive techniques in order to avoid immune surveillance. They are also important in how they would avoid a potential approach from a CAR T therapy. And so the biology is very complex. I think it's very surmountable, and there are many interesting techniques for avoiding or evading the surveillance. One of the obvious ones would be to give it with an anti-PD-1 antibody or give it with a CTLA-4 antibody. Another would be to engineer your CAR such that it can evade these immune suppressive strategies. But solid tumors represent, I think, and Bruce, I don't know if you would agree, but solid tumors clinically represent right now a higher hurdle, a sort of the next big challenge, I'd say, in the clinical application of the technology.
I guess the idea of combining the CAR T approach with other ways to engage the immune system, is it at this point theoretical that the two may be complementary, or is there any data or evidence that would lead us to believe that maybe there are some patients for whom CAR T isn't working because they need some checkpoint inhibition?
Well, there are other things going on in solid tumors. Beyond the accessibility, you can think of the geometry, and in solid tumors you have to peel away like layers of an onion to get at the inside of the tumor. In pancreatic cancer in particular, there is a wall of structural cells surrounding the tumor. So it's harder for the activated T cells, the engineered T cells, to get there. I think combining with the checkpoint inhibitors is something that we'll be pursuing. You can see a recent purchase by Novartis in that direction. That makes eminent immunologic sense. And there are other combination strategies that one can pursue with solid tumors. But again, it is a different beast. It's a more immunosuppressive environment as a whole than the liquid tumors, and there are accessibility issues there, as well as the quality of the raw material, the T cells that you're getting out from the patients, and you may be pulling out in the solid tumors more regulatory suppressor T cells than you do in other types of cancers.
And Josh, there is an expansive now preclinical literature on taking one of two strategies, either engineering the T cell to include other accoutrements or ornaments that will help evade the immune suppression. And then there is also preclinical data directly combining a CAR with an anti-CTLA-4 antibody, for example, demonstrating interesting combinatorial efficacy.
It's interesting to think how the field may evolve and then evolve again and then evolve again and evolve again as we learn so much. Maybe my last question, and then we'll open it up to the audience, and I'll get each of you to take out your crystal ball. And if we were to fast forward to, let's say, the end of the decade, so another 6 years of development, where do you think your field is, whether you define it as CAR T, or Liz, whether you define it more broadly as the gene therapy and Sangamo platform? Where are we in terms of drugs getting their way, or therapies, I guess, getting their way to the market? Do we have multiple drugs on the market across various indications at that point? Are we starting to see the therapies trickle out with a whole host of phase 3? Maybe you can give us a sense of that kind of 6-year vision. Liz, and we can start with you for Sangamo.
Well, I mean, we've got our first gene therapy product approved. And I think that there are, as Jeff said earlier, there are a lot of studies clinical studies ongoing. I think there's an enormous amount of energy and a kind of innovation behind the field. And I think if we certainly will have more drugs approved. We believe that at Sangamo, we're filing INDs in the next couple of years for a number of products in both in vivo and ex vivo applications of our technology. And so my hope is that with no hiccups on the safety angle, which is one thing that could upset the apple cart, that we're going to have multiple gene therapy products, if not approved, then at least close.
So what I find, one of the things I find intriguing is the variety of different approaches here. And I think there is room for all of them done well in the appropriate therapeutic setting. So, we can't have the same hammer, to use your analogy, Josh, the same hammer to hit all the different nails that are possible here. The approach to hemophilia is going to be quite different than the approach to sickle cell disease, which may be quite different than the approach to solid tumors or liquid tumors. The other point that I just wanted to, just coming off of Liz's point and something we didn't hit much upon here, so the Unicure approval in the EU, one of the things that will happen over the next 5 years is more clarity around what the regulatory pathways are. And I think as those of us who are doing the work now begin to sort of blaze the path for how these things end up getting approved in the US And I'd have to say we've had terrific interactions with the regulators here, and they've been very generous and creative with how they've thought about these paths. That will, once there is precedent, once we know exactly how to do it because someone's done it before, I think that will speed things up tremendously.
And I'd like to just echo one point that Rob made. It's a question that we get often is, well, God, what's the regulatory pathway for this? I mean, the FDA must hate you making double-strand breaks in people's genomes. But, it's exactly what you say. I mean, this is a very educated group. They've been extraordinarily generous. They're open to, sort of good conversations about what can be done, and we do the same things that small molecule drugs do, just in a slightly different format. We have to look at toxicology and safety and all of those other things that any drug that's being approved by the FDA. But I have to say that the biologics, and particularly the cell and gene therapy group, are very responsive and thoughtful about what they're asking for us to do. And patient safety is ultimate. We're looking for patient safety and benefit, but it's not the sort of scary thing that people seem to think it is.
So we will have multiple approved gene therapy products. We've begun multi-center clinical trials. Novartis is planning global clinical trials. But I'd also like to pick up on the magnitude of the therapeutic effect. And what that allows is approval based on a relatively small number of patients. And we're not talking about conducting registration trials with hundreds, thousands of patients. When you have the response rates that we're observing, it's going to be a small number of patients. And then the last thing I'll close with is, and we haven't talked much about, is the role of patient advocacy. And I think most people know the role of patient advocates in the HIV, breast cancer community, and in the approval and consideration of Provenge, based on the Provenge data. So can you imagine a delay of approval when we continue to have the therapeutic effects that we do and the response from the patient advocacy community?
Great. Well, let's see if we've got any questions in the audience. Down here.
Hi, the question is aimed at Dr. Levine. This may be a bit more of a clinical question than anything else. I was curious about your comments about the kill switch that you're encoding into your cells. Do you have any idea of how long the cells have to be in circulation before you hit the kill switch? And as a follow-on to that, if you kill, is it possible you need to have the cells, the modified cells, resident for a prolonged period of time to either destroy the malignant clone or to keep it in check? And by eliminating or killing the modified cells, you may, kind of disrupt the apple cart.
Well, that's exactly the risk, isn't it? Even if you can't detect a cancer cell in the peripheral blood or the bone marrow, how do you know that it's not there and that a year later, 2 years later, you will have a relapse? And I think a kill switch is for safety's sake if one observes an adverse reaction. I think there are questions with the safety switches that are out there now as far as the temporal administration of the drug to enable the killing of the T cells. And I can speak to another aspect of safety management that we have employed, and that is there is a particular cytokine, IL-6, that is very elevated during the cytokine release syndrome accompanied by the tumor lysis syndrome. And our physicians have implemented the use of tocilizumab, which is an IL-6 receptor blocking antibody, and that is effective within hours. We've had patients that have been in the ICU intubated, and the ICU nurses can't wean them quickly enough from the pressors, from the intubation following administration of this IL-6 receptor blocking antibody. So we've used that very effectively, and we're also looking at implementing other aspects of that toxicity management algorithm. But I think the issues are just as you say. If you employ a kill switch and you kill these cells because of some potential perceived safety issue downstream, how do you know that you're not going to observe relapse, whether it's months or years down the road.
Do you have any other questions? And if not, I'll thank our panelists for a very enlightening discussion that I think clearly highlights the potential of the future of this platform. Thanks, everyone. I think we're going to move now to our next panel, which will be moderated by Jason Colbert from the Maxim Group on commercialization strategies and pathways to market.